Hi there, this is Mrs. Meek, and we are getting ready to discuss the scientific method. Please be prepared to take notes. Have those notes in class tomorrow. Make sure you identify 1 or 2 questions for discussion. Thank you. All right, so the scientific method is a series of steps that are used to investigate a natural occurrence. All right, so let's review the specific steps of the scientific method. First of all, we have problem and question, observation, research, formulating a hypothesis, followed up by experimentation, in which we collect and analyze results, which we use to create a conclusion, and then lastly, we are going to communicate these results to a scientific community. All right, so the first step of the scientific method involves developing a question or problem that can be solved through experimentation. Right? We want to make sure that we are designing a problem that is something that data can be collected that is quantifiable. We want to be able to generate some numbers. All right, step 2 is observation and research. We want to make some general observation and research your topic of interest. That way you have some background knowledge, and that way you're better prepared in order to run this experiment. Now, do you remember the next step? The next step seems to be everyone's favorite, which is to formulate a hypothesis. A hypothesis is a possible answer to the problem or the question, and in our class we are going to use an if-then statement that will show cause and effect. For example, if soil temperatures rise, then plant growth will increase. All right, after we develop a hypothesis, we are going to develop and follow a procedure. All right, as part of that, we need a detailed materials list, and our experiment needs to be measurable or quantifiable. So we should be able to generate some kind of numbers. All right, step 5 is collect and analyze the results. At this time, you may also need to modify the procedure. All right, you're going to confirm your results by retesting. It is typically it is the norm to run about 3 trials. That way you have good reproducible data. All right, it is also in this part of your experiment that you're going to include tables, graphs, and photographs. Anything that can further support your data. All right, the last step is going to be a conclusion. And that conclusion should accept or reject your hypothesis. All right, you will make recommendations for further study and possible improvements that should be made to the procedure. You will need to use a when-then statement. All right, lastly, you're going to communicate your results to an audience. At this time, you will also take questions from the audience. All right, so let's put our knowledge of the scientific method to a realistic example that includes some of the terms you'll be needing to use and understand. All right, so John watches his grandmother bake bread. He asks his grandmother what makes the bread rise. She explains that yeast releases a gas as it feeds on sugar. John wonders if the amount of sugar used in the recipe will affect the size of the bread loaf. All right, so John has just identified a question that he is going to answer using the scientific method. He does a little background research on areas of baking and fermentation and comes up with a way to test his question. He keeps all of this information in a journal. He then talks with his teacher. She gives him an experimental design diagram to help him set up his investigation, and that might look something like this. All right, after he has identified his question, done a little background research, he then comes up with a hypothesis. So his hypothesis is if more sugar is added, then the bread will rise higher. All right, so the hypothesis is an educated guess about the relationship between the independent and dependent variables. All right, we're going to discuss these variables in the next couple slides because I imagine quite a few of you may have forgotten the difference between the two. All right, do you remember the difference between the independent and dependent variables? The independent variable is the factor that is intentionally varied by the experimenter. So the person that is running the experiment, it's the part of the experiment that they change. So since John wants to know if changing the amount of sugar is going to make a difference, he will use 25 grams, 50 grams, 100, 250, and 500 grams of sugar in his experiment. The dependent variable is the factor that changes as a result of the changes made in the independent variable. In this case, it would be the size of the loaf of bread. All right, his teacher helps him come up with a procedure and a list of materials needed. She also discusses with him the control group. All right, a control group is the group that serves as the standard for comparison. It may be a group that gets no treatment, or it may be a particular group that the experimenter selects. All right, the control group is exposed to the same conditions as the experimental group, except for the variable being tested. So all experiments should have a control group. Otherwise, it leads to poor or questionable results. All right, in this situation, John is going to identify 50 grams of sugar as his control group because that is the amount of sugar his grandmother has always used. John's teacher reminds him to keep all the other factors in the experiment the same, so that way when he analyzes his results, he can attribute that variation to the amount of sugar. So what are some of the other constants that we need to keep the same? All right, can you think of any? All right, they might include things such as bread, the recipe, the oven used, the time that we allow the bread to rise, the brand of ingredients, the cooking time, the type of pan, air temperature and humidity when the bread is rising, the oven temperature, the age of the yeast. All right, so all of these things come into play. So pretty much we need to keep everything else the same. John then writes out his procedure, including a material list, and he has both of these checked by his teacher, and she also helps him to address any safety concerns. All right, trials. John is going to test his sugar variables 3 different times. That means he's going to run 3 different trials. So trials refer to replicated groups that are exposed to the same conditions in an experiment. All right, trials help to validate your data by giving you good, rock-solid results. So John then comes up with a data table that he uses to record his data. He gets his materials together and he carries out his experiment. Here are the results of his experiment. All right, if you look at those, all right, can you determine which group performed the best? All right, so John examines his data and he notices that his control group worked the best, but not significantly better than the 100 grams of sugar. John rejects his hypothesis but decides to retest. This time he uses amounts of sugar between 50 and 100 grams. So his second experiment, he carries out using the materials that he discussed in his materials list. And again, he also runs this experiment using 3 different trials. Here are the results. All right, let's take a look. Can you tell which group did the best? All right, if you look through his results there, take a moment, identify which amount of sugar gave the largest loaf of bread. Hopefully, you came up with the 70 grams of sugar produces the largest loaf of bread. He then accepts his hypothesis, tells his grandmother about his findings, and prepares to present his project in his